Hello friends, in this video lecture we will be discussing a topic from microbiology and the title of our discussion will be mycoplasmas. So mycoplasmas actually are the smallest known aerobic prokaryotes and they have no cell wall. No cell wall is present, so they are known as molecules. Molecules means soft skin, or you can say because no cell wall is present, so they are known as molecules. Although they were discovered by the Pasteur, and when Pasteur was studying the pleuropneumonia in cattle, or you can say the bovine pleuropneumonia, he called them as PPLO. PPLO means pleuropneumonia like organism. You can see over the spelling over here. PPLO. PPLO means pleuropneumonia like organisms. So, Pasteur was not able to isolate them in pure culture or on the standard nutrient media, and he could not observe any property, or you can say he could not observe properly these microorganisms. But then two French microbiologists came, E. Nocard and E. R. Rox. So they were actually this PPLO was discovered by Pasteur, but he was not able to isolate them, or he was not able to procure the pure culture because he was not able to culture them on the standard nutrient media. Then the two French microbiologists, as you can see, the E. Nocard and E. R. Rox, they successfully actually they obtained the pure culture of this microorganism known as the mycoplasma, and having the medium was having the presence of some animal serum, and they observed actually the various forms or shapes of mycoplasma. So they observed various shapes like spheroid shape, thin shape, or you can say the filamentous shape or asteroid shape. Asteroid shape you can see over here, something like a star like. So these kind of irregular kind of shapes were observed by the Nocard and rocks. So because there are various types of shapes, so we also call them as pleomorphs. Pleo means many and morph means form. So they were also observed these kind of pleomorphs in case of sheep, goat, dogs, rat and humans. So then the thus this class was recognized as molecules. As I told you, molecules means without cell wall or no cell wall, or you can say the soft skin. So this order was divided into three families: Mycoplasmataceae. The genus is Mycoplasma with single genus. Echolioplasmataceae, Echolioplasma, and then Spiroplasmataceae with the Spiroplasma. So this is a little bit about the you can say the classification of Mycoplasma or class Molecules. As you can say, there are some stellate forms. Stellate means star-like forms are also seen over here. So these forms, which are actually the stellate form, these are also called as the Asterococcus mycoides. They were also known as the Asterococcus mycoides by the Borel. Borel was a scientist, and Borel gave this binomial term for the species Asterococcus mycoides by the Nocard and Rocks. But Nocard and Rocks called them Mycoplasma. They prefer to call them Mycoplasma. So we have seen this slide regarding some of the characters of Mycoplasma. Who discovered them? Louis Pasteur discovered them. Then the Nocard and Rocks obtained them in the pure culture. The size of microbes 0.1 to 0.0 0.25 micrometer likewise. Okay, so this slide is for actually the classification of bacteria given by the Burgess Manual of Determinative Bacteriology. So I just want to tell you over with the help of this slide that if we classify the bacteria as per the Burgess Manual of Determinative Bacteriology, there are 19 groups, and you can see over here group 1, group 2 likewise, and in the 19th group mycoplasmas are there, right? So this is the position of the mycoplasmas as per the classification of bacteria given in the Burgess Manual of Determinative Bacteriology. So the purpose of including this slide in this presentation is just to tell you that where is the place of mycoplasmas in the Burgess Manual of Determinative Bacteriology, right? So let us see that Schizomycetes is also divided into 10 orders on the basis of morphological characters and motility. We can see the name of orders here, over here one by one. Pseudomonadales, Chlamydobacterials, Hyphomicrobials, Eubacterials, Caryophenales, actinomycetales, veggiotales, myxobacterials, spirochetes, and myxoplasmatales. So I would like to tell you just one thing over here, that if we classify the class Schizomycetes, there is a 10th order which is known as the myxoplasmatales. Okay. So now let us discuss about the salient features of mycoplasma. So if we discuss about the salient features of mycoplasma, we can see over here that they lack a true cell wall. So no cell wall is present. They are actually the unicellular organism. They are prokaryotes and they are non-motile. No motile structure is present over there. And if we talk about the nuclear content or nuclear material, then both DNA and RNA are present. You can see over here, I have written over here, both DNA and RNA are present. But the amount of DNA and RNA is very less in compared to the other prokaryotes. You can write over here that DNA is there about 4% and RNA is 8%. So we are talking about the DNA and RNA which are present but in very less amount, you can say. The size is also from very 1 micron to few micron, and that means they are very small, and the shapes we have already discussed, and their reproduction is by budding or the fission of cells, binary fissions of the cells, as I given you the information prior that they have no flagella. No flagella is present, no spores. They are gram negative. They are gram negative. We will discuss in some other presentation regarding the gram staining. On the basis of staining, we describe the bacteria into two types, that is gram positive and gram negative. But here we are not going to discuss all those things. We are just categorizing the mycoplasmas as gram negative. Okay, so they are parasitic. Now look here, they are the parasitic on humans, animals, and they are also saprophytic. And when we grow on these mycoplasma in not artificial nutrient media, that means if you are growing them on some nutrient material, or you can say some medium having some nutrients added to it, then they produce the colonies, and the colonies are in very minute form, but a particular characteristic feature, or you can say a particular appearance, is that which is known as the fried egg appearance. So they give the appearance of a fried egg if they are cultured on some nutrient media. So they are also isolated from the healthy and diseased animals, humans, and they are suffering from various diseases. If we just talk about the uh, action of antibiotics on the mycoplasma, then we can say that completely resistant to penicillin, because they don't have any cell wall, so they are resistant to penicillin, but they are sensitive to tetracycline and chloramphenicol. So you can include three antibiotics in your description, just to tell about the resistance and sensitivity of mycoplasma. So penicillin, to the action of penicillin, they are resistant because penicillin acts on cell wall, so they don't have any cell wall, and they are sensitive to tetracycline and chloramphenicol. So let us move to the next slide. Okay, so now we are going to discuss about the mycoplasmal diseases which occurs in human being. So I have marked over here that basically the mycoplasma, they affect the you can say the respiratory organs, or the CNS, that is central nervous system, and cardiovascular system, urogenital system in human beings. And those species which are more extensively studied in case of human beings causing infection, these are Mycoplasma hominis, which is causing pleuropneumonia, inflammation of genitals, and endocarditis. And the Mycoplasma pneumoniae, which is the, you can say, the species of Mycoplasma, which will cause the atypical pneumonia. In case of Mycoplasma fermentans, they cause the infertility in humans, or you can say in man. And the Mycoplasma oral and salivarium, they also found in association with the human beings. So you can see that we have discussed a few species of Mycoplasma in reference to the human beings. That means the Mycoplasma is also capable of making the human beings, some kind of diseases can develop in human beings, and they can be the species can be Mycoplasma hominis, Mycoplasma pneumoniae, Mycoplasma fermentans, Mycoplasma salivarium. So you can just go through the slide and you can note down some of the species and some of the diseases as we have discussed. Okay, so now we'll move to the next slide. Now we are going to discuss about the mycoplasmal diseases in animals, right? In the previous slide, we have discussed about the diseases occurring in the human beings. Here you can say that mainly the mycoplasmas are actually isolated from the cattle, or you can say goat or sheep likewise. So mycoplasma mycoides causes pleuropneumonia in cattle, and some of the species they cause the pleuropneumonia in goats. Mycoplasma bovigenitalium cause the inflammation of genitals in animals, and Mycoplasma agalactia was isolated from the sheep and goat. So these are the few species in case of mycoplasmal diseases in animals, and especially in reference to the cattle. So you can note down the name of the species as well as the disease, or you can say the symptoms. For example, the inflammation of genitals, likewise you can just note down. Mycoplasmal diseases in plants. Now in this slide we are going to discuss about the what are the name of the diseases, what are the symptoms and vectors, especially in the case of plant. So you can see over here darkness, loss of apical dominance, enlargement of floral buds, bunchy growth of newly formed sprouts. So these are some of the, you can say, the symptoms which occur in the plants if the mycoplasmal diseases are there in the plants. So these are the a few symptoms which actually arises due to the disbalance in the plant hormones which occur due to these mycoplasma. Now, how these diseases are transmitted? So, the transmission of these diseases occurs by the vectors, which are the insect vectors. So, insect will transmit these mycoplasmal diseases belonging to the class Psyllidae. And the multiplication of the mycoplasma, you can say, it will occur inside the insect body, and when this insect will some other plant, so it will transfer the uh, mycoplasma. And some of the mycoplasmal diseases of plants, you can say, that is little leaf disease of brinjal, clover daft disease, then phylloidy, bunchy top of papaya, which is bloom of legumes, stripe disease of sugarcane, and yellow dwarfs. So you can note down these are the name of the diseases which I have underlined: clover draft, phylloidy, bunchy top, stripe disease, and these are the few symptoms of the mycoplasmal diseases, and these are the actually the vectors which belongs to the insect category. So these are the vectors Psyllidae, and these are the some insects which are the responsible for the transmission of mycoplasmal disease. So you can see the shape of the mycoplasma over here. You have to include this diagram in your answer to just make the various shape. And this is a single mycoplasma where you can see that no cell wall is present, but lipoprotein membrane, three layers are present, and ribosomes are also present, soluble proteins are present, and DNA and RNA is also present, which is present in the, you can say, very less amount in comparison to the other prokaryotes. So in nutshell we can say that these mycoplasma are actually the smallest known aerobic prokaryotes. They are without cell wall, so they are more known as molecules, and they were first discovered by Pasteur. But Pasteur was not able to isolate them in pure culture, and he did not observe them properly. So then again, the two French microbiologists, E. Nocard and E. R. Rox, they successfully obtained their pure culture, and they obtained their pure culture in a medium having the presence of animal serum, and then they observed these kind of, you can say, the various type of shapes. So you can just write down the order and families of mycoplasma, then the position of the mycoplasma as per the Burgess Manual of Determinative Bacteriology. Then you can just tell in which order they belongs to, some of the salient features of the mycoplasma, then the mycoplasma diseases in human beings, in animals, and in plants. Thank you.